Hello, I'm Jim Cornell, and this is Base to Base, the weekly biotech podcast from Air Coastal Media. Yes, welcome again to the Base to Base Biotech podcast, and this episode is going out on May the 23rd. Another long weekend coming up in the UK, for some at least. As always, I forgot and scheduled 2 interviews. Still, the forecast is rotten, so I'd likely not be doing much anyway. I guess it would be described as dreich in Scotland. It's a whole new language. Before we get going, if you have any suggestions for who should be on the podcast, please do get in touch at jim@deeptechdigest.com. Now comes my memory test as to who was on the podcast last week. Bit more of a test this week because there were 3 guests on it. It was a show dedicated to companies that attended the AACR. And those 3 guests came from Microbiotica, Tessellate Bio, and One Cell Diagnostics, which is now called OneCell.ai. I mentioned last week that we did have a 4th interview with another company that had presented at AACR in Chicago, but the file arrived just a little too late to edit in time for the podcast. The great thing is there's always another podcast coming right behind, so we can run it this week instead. And so that's what we'll do. So this week we have a chat with AOA Dx CEO and co-founder Oriana Papin-Zoghbi. But first we have a really interesting conversation with Vidmantas Šakalys, CEO of bioprinting company Vital3D Technologies. As well as developing 3D bioprinting of organs for transplants, the company is also looking at bioprinting because of its promise in cancer treatment by producing tumour models from patient-derived cells. With cancer on the rise globally and the mean cost of cancer drug development estimated at $4.4 billion per drug, bioprinting could alleviate some of the many issues currently affecting the healthcare system. So I guess maybe to start then, if you could tell me a little bit about the company.
Yeah, so Vital3D Technologies is a kind of young startup, 3 years old. For startup, maybe it's not young anymore, but for deep tech startup, it's still in the early stages. Currently 10 people. We managed to raise a $2 million pre-seed round. And for this, our target was to develop the technology for bioprinting large-scale tissues, and we especially targeting the kidney as transplant, to print the kidneys as transplants. And for this initial investment, we already have a laser-based femtosecond printer, bioprinter, was developed from the ground for usage in clean laboratories. At the moment, we already done with technology development, and now we are working towards the tissue printing.
How does that work? How do you print human organs?
Yeah, so actually, bioprinting is not a very new concept. It's known from late 80s. The thing is that the field is really challenging. A lot of challenges to be solved. One of the key challenges that we are solving is printing the vasculature network. And for vasculature network to print, actually for large-scale tissue, vasculature is a must. Otherwise, the cells just simply die without the proper nutrition reaching every cell, so to say. So for vasculatures to print, you need both precision and speed. And current technologies, we are only precise or fast, but not both at the same time. And we are developing technology to do both of these things. Be precise enough to print the smallest vasculature, that's about 3 microns in diameter, and be fast enough to be able to print the whole kidney in like 48 hours. That's about 150 kilometers of length of overall vasculatures in one kidney. So how do we print? We use the laser-based printing, and laser-based printing is actually using photosensitive polymers that gets hard under the light. So in fact, it's like a gel substance, but when you shine the light into it, you harden it. So what do? We mix the polymer with cells, so you get so-called bioing. And when you shine the light laser like a pencil through all the amount of polymer gel, and you harden it by bit. So when you finish printing, you wash away residual gel, and then you have the structure. It's like scaffold that contains the live cells that are kind of attached to the scaffold itself. After this process, you just leave the scaffold with the cells in bioreactor, and then the tissue starts to grow. So it's, in essence, bioprinting is from two parts, printing and growing, so to say.
What's the origin of the cells? Does it have to come from the person that you're potentially going to be transplanting into, or can it be from any source?
So actually, yes, currently we experiment from any source, so to say, but our target is to have cells from the patient to whom the transplant is printed. So the idea would be to use the patient cells to print transplant for him, avoiding these immunoreactions that usually just kill the transplants, not allowing them to fully adapt to the patient body, so to say.
Would there be any, obviously all organs are different, but would there be any rejection issues, and is there a way to overcome that in the printing process?
In fact, we are still far away from live, like implantation. I think it will be rejection issues, but if we use the patient cells, it will be much less. The thing is that currently the big challenge is to understand how the cells are growing and how you can manipulate the cell types, because you don't want to use 20 different cells in the printing, because the kidney, for example, needs 20 different cell types for functioning. So it will be very challenging engineering thing if you print from all 20 different types at the same time. So the target is to use much less cell lines, maybe stem cell lines, that you can differentiate one or another way into the different type of cells, vascular cells, muscular cells, all the cells that are needed by the kidney. So there are still a lot of challenges to be solved regarding the cells itself.
As far as the organs themselves are concerned, which organs do you believe will be the best ones that can be printed, and are there any that you think may not be able to be?
So actually, the organ that is currently printed in some amounts and already used in cosmetic industry for avoiding the using animals for testing is the skin. Skin is easier to print because for some applications you even don't need the vasculature. You just print the thin layer of the skin and feed it from the both sides. And then you can use it for cosmetic testing, allergies or something like this. So there are several patents already on skin. Skin is still not used on humans because, the usage on humans requires a lot of formalities to be passed. But I believe the skin will be the first organ that will be used for burns or diabetes or something like that. And it's easiest to do. The hardest, the kidney is one of the hardest organs, but I think, if we omit the brain as such, we don't want to print the brain, yes. If we omit the brain, then the hardest is the heart, actually, because the heart cells must withstand quite strong electrical impulses and beating like this. So that means that the printed tissue has to have very strong resistance to mechanical manipulations, and it will require quite a challenge to solve it.
How do you ensure that the cells are viable and that they are both alive and stay alive before you can perform the transplant?
Yeah, so for cells to stay alive, you need to feed them somehow. So if you print the like thin layer of the skin, you can like immerse into the specific liquid, feeding the cells like that. Our target, as we are working on the skin as well, because the skin is easiest organ to print, and we see it as a first step from our side as well into the printed organs. Our target is to bioprint the vascular network for skin and start feeding through the vascular network the skin. So we are targeting more complex skin model, so to say.
Right. And moving away from the transplantation side of things, how can printing of organs or parts of organs help with research and drug discovery?
Yes, so one of the earliest wins in this area could be drug testing, drug discovery. And for that, I think the cancer treatment is the target that we can approach at the beginning, because with a 3D dimensional printing, you can print the specific tissues and experiment with the drugs, leading to like drug discovery, faster drug discovery, and drug testing. For example, our experiments and our printed scaffolds that we are currently working with National Cancer Institute allows organoid formation from the cancer lines that usually do not like to form the organoids, meaning that if you use only cells, we are not grouping into organoids, and you cannot drug test these cancer lines because it's very difficult to test on a separate cells. But given our very small scaffold, these cancer lines start to grow into organoids, and Lithuanian scientists are already testing on these lines the drugs for cancer.
What about the economics of this? Would it be economical for drug companies to be able to use printed organs?
I think yes. That's our target, actually, to make this precise printing of organoids available for mass usage. Really, our technology has quite good possibilities to be speeded up to such a point that printing organs for drug testing, even for maybe surgery, so to say, training for students printing some organs, it could be a viable option with our technology. So that's our target, to introduce this technology to wide use.
And as far as testing drugs on printed organs, would that be something that you can guarantee the reliability of the results?
So in drug testing, actually, the big challenge is not to just test the drug on specific organ. The big challenge is to understand how the drug affects other organs in a human body. So for reliable drug testing, actually, what do you need? We need a human on chip, meaning that we need to combine different organoids from different organs into one chip system where you can like reliably test the drug toxicity on different parts of the organisms. And that's with the bioprinting, like human on chip system is really achievable.
What would some of the other advantages be to using printed organs for research?
First of all, we omit the usage of animals in research. Everyone wants to kill less rats, less rabbits for common good. So with bioprinted organs, this possibility is really high, that printing organs for research will reduce reliance on animal testing, very heavily reduce.
Are there any other challenges? You mentioned the effect on other organs and having to have other organs involved. Are there any other challenges or limitations?
So yeah, the ethical challenge of accepting that the body could be a challenge in different cultures where the people are differently treating live cells versus the challenge of regulation is quite big as well, because nobody knows how to treat the printed organ. Currently, we have either medical devices or drugs in regulation, and printed organ is something in between. It's not the medical device because it's live thing, but it's not a drug actually. So yeah, we need to set the guidance how we will regulate this topic as such.
Has that already started? Are there any discussions on that yet?
Yeah, it's only initial discussions currently going on, and yeah, all the printed things are like more like medical devices, not some drugs, but we need to understand how it will move forward. Only starting discussions.
Are there any other companies doing anything similar in this area?
Yeah, actually, as I said, bioprinting is quite old concept, and there are a lot of companies and academia groups that are working on completely different topics because there are a lot of challenges in this area. In my opinion, the market currently is just rising, and all the companies that are working in this field are kind of helping to understand and to grow the market together. So kind of creating the market field for growing tissues and organs for different applications.
Are you having discussions already with drug companies about the way that this relationship could work with them?
Yes, we are meeting and we are discussing with drug companies, with more smaller contract research organizations that are working with drug companies. So we are looking in a ways how we can capitalize on these discoveries and how we can move forward with drug testing. Actually, because we need we are interested for the bioprinted products to go into a market faster. We are developing our own patch, currently only patch for treatment of dog wounds. So we are approaching veterinary market that is not so heavily regulated for testing the approach, testing the technology, validating the products. So because the dogs that are usually old ages, like over 10 years, they like 50% of them get cancer, and like one in 200 of dogs needs some surgery operations. During the cancer, there are tumors that are operated, and especially on the legs for dogs, there are not enough skin to cover the wound. And these like wounds, they take like 5 months to heal, and that's burden for dog, for owner, for everyone. And our target is to like build a patch that will kind of cut the healing time in half at least. So restoring the skin in these areas. So this way we kind of test the technology, and then we will move into the human applications after this veterinary field.
And so what are the next steps for the company? Where do you go from here?
So yeah, from this we go to the human patches for like things like restoring skin after diabetes, after burns, and then later on we will move into the sophisticated skin models and repairing like even internal organs with different internal patches. And from that, we will move more forward to something like our very complicated skin and internal organs bioprinting, so to say.
All right. Do you have a potential timeline for that? I know it's difficult to say, but
yeah, for the overall like first organ to be printed, everyone agrees in the field that there are no principal obstacles, but the timeline is like everyone projects that 10-15 years we will see our first organ bioprinted.
That's not that far away.
Yeah, it's not very far away, actually.
Yeah. Those are the only questions that I had. Is there anything that we didn't talk about that you wanted to mention?
Just want to say that actually in Lithuania we have very good ecosystem. In Lithuania we do have all the three fields that are essential for us, very strong. So lasers, biotech, and information technology, and we use all these in what we are building. It's all these three fields. So for us, it's kind of we are building it on kind of good ecosystem that exists in Lithuania and good knowledge in three different fields, creating the innovation on cross of these fields, so to say.
I actually think bioprinting and 3D printing in general is quite fascinating, and as the technology improves, surely this is an area with great potential, not only for transplants but also for research. Printing has come a long way. Next, we're looking at the development of a test that can detect ovarian cancer early, which is a huge step forward in saving lives, as more than 70% of women diagnosed with the cancer are diagnosed at stages 3 or 4, when the 5-year survival rate is less than 30%. So to tell us about this great leap forward is Oriana Papin-Zoghbi, CEO and co-founder of AOA Dx. So I guess first then, if you could maybe give me a little bit of the history of the company.
Absolutely. I'll start a little bit before the company. My co-founders and I, we've worked together for 10 years prior to starting AOA, always at early stage startups focused in developing diagnostics and with a focus in women's health, and really how do you bring to market innovation that changes the clinical pathway. We worked from the early days of those companies all the way through commercialization, both companies going on to strategic exits. And after the second exit, the three of us got back together again and said, how can we continue to have a really big impact in women's health in particular? And we're not scientists by education. And so none of us were going to be the ones that were going to take our PhD work or, a great scientific idea and go ahead and develop it. We always knew we came at it from the market need side and really built a model around where are there big areas in the clinical pathway where we're missing good diagnostics, such as ovarian cancer, such as endometriosis, such as preeclampsia, and how can we partner with incredible researchers and academics and innovators to help bring some exciting scientific invention into, the development of a product. So we ended up partnering with a professor at McGill University, Professor Šakalys. This was in late 2019. We were introduced to him. And we took a leap of faith with his early research and applied for some non-dilutive funding and got some grant funding to get us started. And in 2020, essentially ended up founding the company around the idea that glycolipids could be novel biomarkers for the early detection of cancer. And we were going to be the ones to build that into clinical diagnostics.
It seems to be an unusual way to start a biotech company. Not that there's anything wrong with it. It makes perfect sense, but it doesn't seem like it's the usual model.
Yeah, I think we pride ourselves at AOA of being unusual. Like, how can you attack the problem in an out-of-the-box way? Just because you don't have the scientific background doesn't mean that you can't have an enormous impact in this space. Doesn't mean that you don't need that expertise. You absolutely do. The first thing we did when we were able to get venture funded was hire a rock star chief scientific officer because we knew that was the missing piece of our puzzle. But I think what sets us apart, particularly after the 2020, 2021 sort of boom in financing and every possible wonderful idea, is like what keeps us alive and thriving in 2025 is that we have a really solid business model on how do we take science and technology into product and market. And I think that's what sets us apart.
I think, though, when you look at it from the opposite angle, quite often scientists don't necessarily have that business acumen. So it kind of works both ways. It's like if you're a business person partnering with science, it's no different to the scientist that has to bring in or learn the business side of things.
Yeah, it's yin and yang. It's having the humility to say we don't know it all, and how do you bring the right people to the table to make the strongest team possible?
What have the challenges been so far in developing tests for early cancer detection? And I don't necessarily mean just for your company, but in general?
Yeah. In general, in this space, and I'll narrow the question down to like liquid biopsy, right? Because there's a lot of in early cancer detection, we have imaging options, we have different types of techniques, different types of samples. But if we talk specifically about blood samples, one of the biggest challenges we faced is how do we diagnose the cancer when it's still in the earliest of stages, so stage 1 and stage 2, through a blood sample. And to summarize, one of the areas of challenges has been blood-based biomarkers don't necessarily have the sensitivity to pick up the disease when it's still very, very early. And the industry overall historically has focused on each company or each innovation is working on a single biomarker class. So you have those that have become experts in building products on circulating tumor DNA, those that have become experts on proteomics, those that have been experts on, exosomes or microRNA. And what's changed in the last 5 years is this concept of multiomics, and how do we start to combine different biomarkers together to really help solve that problem of can the combination of biological data help us uncover those early stage cancers through a blood sample?
And so how did you kind of go about overcoming some of that and getting to where you are?
Yeah, 2 steps. The first thing is we started looking at an entirely new biomarker class. So of all those that I named, we didn't start there. The work that Professor Šakalys began and what got us so excited about founding a company was that this was something novel, new, and exciting that had the impact to be groundbreakingly beneficial to the cancer community. And that was lipidomics. And so we started by using his early research and continuing to prove out the hypotheses that lipid-based biomarkers could be really valuable in cancer detection. We then took that one step further in using this multiomic approach that I mentioned and saying, okay, if we look at it, instead of lipids alone being the one biomarker that can really help solve this problem, what if we combine these novel lipids with proteins that are somewhat tried and true in cancer diagnostics? What is the power of bringing those two together and using machine learning and the advances in that to really help increase the predictive power of early detection and cancer diagnosis? And so that's the approach we've taken. We've taken a really novel biomarker class and we've innovated there, and we've combined it with protein biomarkers, and we've developed a machine learning approach that allows us to really hone in on being able to diagnose ovarian cancer.
Is it an area that many other companies or researchers are working on?
In terms of multiomics, absolutely. We're not the first to say, the power of combining biomarkers is going to be really great. In terms of really tackling ovarian cancer, we're the only ones doing it as uniquely as we are, which is that we're looking at women that are symptomatic of this disease. I think others in the ovarian cancer space have tried to innovate in population-based screening. Or, for example, once a woman already has a mass on imaging on triaging that mass, which is 2 different ends of the spectrum. Where we're uniquely focused, when there really aren't any other companies doing what we're doing, it's focusing on when a woman is presenting to her doctor with vague abdominal symptoms, how can we accurately rule out ovarian cancer incredibly early, avoid a very lengthy and costly diagnostic odyssey, and instead focus on those few women that have the potential of ovarian cancer and catching them much earlier?
Obviously, we're talking about ovarian cancer, but does this apply to other cancers as well?
Absolutely. The methodology, the technology, the approach can be used in various other disease areas as well. So from our academic work and academic partnership, we published papers on where the potential of these biomarkers may lie in other cancer indications as well. And that's something that we're incredibly excited about exploring. The other area for us to explore is within our own biobank. So we've been prospectively enrolling patients in the United States that have vague abdominal symptoms. As you can imagine, with vague abdominal symptoms, the final diagnosis is quite varied. Some women will have ovarian cancer, and some women will have other cancers or other diseases. So we're very eager to explore the potential on our platform, even just within our own biobank, and understand how can we develop signatures for the other diseases and start to differentiate from all these different things that a woman could have when she's presenting with vague abdominal symptoms.
Yeah, and the hope is always that it's something completely trivial, so that's always a good thing.
Absolutely. I mean, the best case scenario is that it's not ovarian cancer. And quite frankly, for the majority of women, it won't be ovarian cancer. The challenge is a lot of these women that don't have ovarian cancer end up on a cancer pathway, which is huge anxiety, but also very costly. And on the reverse side, for the women that do have ovarian cancer, are on a pathway that is so long and dragged out that is preventing an early stage diagnosis. So in the United States, the average time from when a woman presents to the doctor with symptoms to when she actually gets her diagnosis is 9 months. Early stage tumors double in size every 4 months. So that tumor is spreading so quickly just while she's stuck in the healthcare system, not knowing where to triage her, how to get her to the right next step. And that's incredibly challenging. If we could get the women to the right next step much faster, we have the potential of stage shifting this diagnosis and catching it much earlier. And that has the life of an impact when it comes to 5-year survival, right? 80% of women today are diagnosed stage 3 and 4. Their 5-year survival is 28%. 28% for the majority of women diagnosed with ovarian cancer. Versus those lucky few that are diagnosed early, their 5-year survival rate is 90%. That's with currently existing treatment modalities today. So if we're able to stage shift some of these diagnoses, we have such a huge impact to actually improve the outcomes of this disease.
Do you envisage that, I'm kind of going off topic a little bit here, but or maybe this won't even work, but at some point, do you envisage this being something that could be like an annual test, or that you don't necessarily have to go in with symptoms, that you just test people regularly?
There is a world where that can happen. There's a lot of things that we need to consider when we think about population-based screening. Number one is the incidence of the disease, right? One of the challenges with ovarian cancer is it's a low prevalence disease. Very few women in the world actually get ovarian cancer. And so you have to think about that from the effects of a cost mechanism of how many women do you have to screen to find the needle in the haystacks of the very, very few. So in that context, what we're aiming to do is understand what is the performance of our technology in a population basis, coupled with what the cost will be. And if there is a viable accuracy as well as a viable cost, then it makes all the sense in the world. We now know that we believe we have the accuracy side. At AACR, we presented 2 sets of data on the symptomatic population and data on the screening population, both showing incredibly high accuracy. So from a technology perspective, we have a lot of confidence we can achieve that. The next step is understanding the health economics that's different in the US as it is in the UK, as it is in other countries, and understanding how a test like this could fit in for screening the population. But I definitely see that there is a potential for that in the future.
You mentioned the AACR meeting. How did your presentation go down there? What was the reaction like?
Oh, absolutely incredible. We had 2 oral presentations, which were flooded the entire time. My chief scientific officer, Abby McElhaney, and one of my scientists, Rachel Culphill, were both there, and they did not have a second to breathe because people were approaching them from industry, from academia, researchers. We got approached for partnerships on the concept of multiomics and said, wow, you have some really interesting data. What if we partnered your biomarkers with our biomarkers? How could that continue to help? So we were really overwhelmed by the positive support on the data and on the approach as well. I think what people are really excited about is that this is different. This is not reinventing something that has already existed in the space for some time. You know, lipidomics is an up and coming field, and AOA is at the forefront of that. And, you know, people get really excited when they see something new in the industry. And then also with our data, we were showing over 90% AUCs, area under the curves, in all of our populations, which is really exciting when you think about the diagnosis of ovarian cancer.
Going back to some of the things that you do, could you tell me what glycolocate is?
Yeah, absolutely. Glycolocate is what we've named our platform. And so think of AOA as sort of 2 steps. There is the discovery platform, which is really trying to understand what are the right biomarkers and what are the combinations of biomarkers and what does the algorithm look like to identify the right whatever product category we decide to go after. The first one is called Acrivis, which is for ovarian cancer. So Glycolocate is our platform that allows us to do all of our discovery work. From there, we spin out products that are specific to disease areas we decide to work on. Our first is ovarian cancer. That product is called Acrivis. That is a product for the diagnosis of ovarian cancer in women with signs and symptoms. And then out of the Glycolocate, we can spin out products 2, 3, and 4 for other indications as well. The focus of Glycolocate is really understanding discovery biomarkers across both lipids and proteins and potentially other analytes in the future as well. Whereas the focus of the products is to narrow them down and really build signatures for specific disease areas.
And obviously, you mentioned Acrivis. Are there other ones in the pipeline or already started? Or
There are other research opportunities in the pipeline. We haven't got as far as naming them yet, but there are certainly other indications which we have our eye on to pursuing. We're also in discussion on collaboration partnerships, so where we work with other organizations, be it pharma companies or diagnostic companies, to explore new indications as well. So we certainly do have a lot of opportunity in the pipeline.
well, as if the one thing that you're doing now, as if that wasn't enough already, keeps you quite busy.
It keeps us incredibly busy. And I think, one thing that changed in the last 5 years was the financing landscape. And the financing landscape today is incredibly focused on getting a product to market and really driving revenue and growth. The way that we view it is that the way we prove glycolocate is by having a product work on the market. Otherwise, we're publishing papers and doing research, and that doesn't actually advance the field in terms of, patient care. And so for us, if we can prove the first one can get to market effectively and actually impact patient care, then we know, that glycolocate will be immensely successful.
Speaking of which, what is the timeline for getting this available?
We're pursuing an FDA pathway, and we estimate having our approval process and being able to launch this product at the end of 2027. So in about 2 years.
Very good. Yeah. And what does early detection mean for patient outcomes? Because I know you mentioned the stages and 90% figures.
Yeah, it has a huge impact on patient outcomes. So if we're effectively able to stage shift these diagnoses and diagnose these women at stage 1 and 2, their 5-year survival rate is 90%, and their recurrence rate is only 14%, which is incredibly good, versus what the world looks like today, which is these women primarily are diagnosed stage 3 and 4 that have a 5-year survival rate of 28% and a recurrence of 96%. So these women are reoccurring, they're going back for treatment, and most of them don't make it to 5 years.
Not just the financial side of things, but the emotional and the effect on families, and it's a huge issue.
Absolutely, it is. Honestly, on both sides of the journey, whether you go through the pathway and find out at the end of the day that you do have ovarian cancer, obviously that's incredibly hard on the individual. It's incredibly hard on their family. It changes the landscape of their life now because they are going to be in treatment and they know that they're likely going to reoccur, and they're waiting every day to know, is it back or is it not back? What chance do I have? And I've heard this story on multiple occasions, both from people that I've met in the field, but even by family and friends who have had close individuals, diagnosed with ovarian cancer. On the flip side of it, because of what the diagnostic odyssey looks like today, you have women that are going through undue anxiety because they've been put on a cancer pathway to rule out ovarian cancer. They're having very invasive procedures only to find out at the end of the day that they didn't have ovarian cancer, which of course is such a huge relief. And in the grand scheme of things, that's the preferred outcome. But can we avoid some of that anxiety? Can we not send women to oncologists if we don't have a higher level of confidence that this could be cancer?
What kind of timelines do you have for your product in terms of testing someone and them being told, yes, you have it, or no, you don't?
Yeah. So the pathway that we're looking at today is when that woman comes in right at her first appointment, she's complaining of these symptoms. Typically, clinicians will do some sort of a workup. Usually it involves a pelvic exam and some type of blood work. They don't send to imaging right at that first appointment. And then typically is they have a wait and see if your symptoms are persistent, track your symptoms according to your cycle, try some over-the-counter medication. And if you see that these symptoms are really persistent, come back again in 6 to 12 weeks. And then, at that point, they'll consider doing some imaging. And then at that point, depending on what they see on imaging, they may refer to oncology or they may not. They may continue to see, let's see if the imaging changes, come back again in 2 months, we'll do another scan. It's quite dragged out. What we're looking at in our pathway is that woman comes in, she's getting a pelvic exam, she's getting some blood work, they draw for our test. They have an Acrivis test. We're still working on the final, what the report readout will definitively say, but imagine it says high risk, low risk, positive, negative, something like that. Let's say it's positive. Okay, this woman has an elevated risk of ovarian cancer. Send her to imaging right away. Not this, come back again in 6 to 12 weeks and see if your symptoms are persistent. It's no, go to imaging right away. And then based on that imaging, you can send her to oncology right away. And so what we're trying to do is give clinicians a much stronger level of confidence on, okay, these are the women that we believe are at risk of ovarian cancer because we have a biomarker, we have imaging, we can send these ones to oncology much faster. And we've eliminated all of these ones, with a negative predictive value of 99%. we can confidently rule out ovarian cancer in all the women that have a negative test and say, okay, we really feel confident that these are not women that need to go down a cancer pathway, and we can likely work them up for something else because they do have symptoms and there's likely something wrong, but it doesn't need to be through an oncological pathway.
As well as the psychological side of things, there's a good economic case to be made as well, insofar as you're not doing unnecessary tests or spinning it out over months.
100%. There's a huge economic incentive here because, number one, you're not doing unnecessary tests. It's not spanning over months. On average, in the US, women see 4 different specialties before going to an oncologist and eventually getting diagnosed. Can you imagine the costs that are building up over time? But on the flip side of that, the majority of women being diagnosed late stage is hugely expensive. Treating late stage disease is so much more expensive than treating early stage disease. And ovarian cancer is the second most expensive cancer to treat. It's second only to brain cancer. So it is costing the healthcare system an enormous amount of money to identify which of the women that are presenting with symptoms actually have ovarian cancer or have a high potential of having ovarian cancer, and focusing our energy there quickly rather on a long pathway of so many women and trying to find those amongst them.
I imagine you probably, you were saying 2027, I imagine you wish it could be next week, but...
The number of times I've had people reach out on our website, reach out on our social media, and that have said, I want this for my mom, I want this for myself, how can I get involved? Sometimes the best we can do is we say we have an active clinical trial. Please join, the clinical trial if you're interested in being a part of our research. But it is something that I wish I could get out there much sooner. At the same time, we always want to do our rightful process, right? We want to make sure this is safe for patients. We want to make sure this is safe for providers. We want to make sure we build the evidence for payers to reimburse this so that it doesn't have to be an out-of-pocket test for women. And those things take time, but the outcome will be well worth it.
A terrible disease with not great outcomes, so early detection. That's a terrible disease with not great outcomes, so early detection of ovarian cancer can make a massive difference to patients. Anyway, that's it for this week's podcast. If you do have questions or comments or suggestions for guests, please do get in touch, and you can do so at jim@deeptechdigest.com. So, until next week, wherever in the world you are, I hope you have a great week. Thanks for listening, and I hope you'll join us again next time for another Base to Base Biotech podcast. The Base to Base Biotech podcast is an Air Coastal Media Limited production.